Good evening to all the So here we are in our last session of Phycology, and we are also going to begin Mycology today. So here we are left over with the topic that is Cyanophyceae, which are the primitive algae, primitive algal members. If you look into the schematic position, the family Psychophyceae belongs to the order Nostoceales and the class Cyanophyceae. Okay, and division is Thallophyta. Okay, the division is Thallophyta here because it also belongs to the algal members. General characteristics of Cyanophycean members, if you see, the habitat, firstly, the occurrence or habitat of Cyanophycean members, they usually occur in ponds, lakes, paddy fields. Mostly they are associated with moist surface regions, and few of them will also act as symbionts. For example, nostoc in Anthoceros and Cycas, Anabina in Azolla. Okay, the symbiotic association, main purpose of the symbiotic association is nitrogen fixation. Nitrogen fixation is the main purpose of this symbiotic association. Clear, right? And next, if you see, thallus organization. Cyanobacteria are Cyanophycean members, where we also call them as blue-green algae. Okay, we also call them as blue-green algae. These BGA members can be unicellular, multicellular or filamentous. They can be unicellular, filamentous, multicellular forms also. Unicellular, filamentous, multicellular forms. Any of them it can be. So here if you observe, thallus unicellular or filamentous it is. Filaments are unbranched in Cyanophycean members. Okay, Cyanophycean members, filaments are unbranched, and filament, each filament is addressed as a trichome structure. What is a trichome? A single row of cells is addressed as what? Trichome structures. Okay, these trichomes are surrounded by mucilaginous sheath, which is often gelatinous. Remember, filaments are usually unbranched. We address them as trichome structures, and covering these trichome structures there is a gelatinous sheath. Here we go with cell structure, prokaryotic cell structure. Cells lack true nucleus. Genetic material is nucleoid region. Genetic material is present inside the nucleoid region. And here see, cell structure is prokaryotic. So there is no well defined nucleus in the cells of the cyanobacterial members. Each cell will have a double stranded circular DNA is present. Double stranded circular DNA is present, which is not uncovered by a double layered nucleus. It is just present at one region, situated at one region of the cytoplasm. It is not scattered into the entire cytoplasm, but this is situated at one region, which is nucleoid. This is nucleoid. Fine, nucleoid region. There are no plastids, mitochondria or endoplasmic reticulum and Golgi bodies here. Cell wall, where inner cell wall is made up of peptidoglycan and outer is mucilaginous. Okay, inner cell wall is peptidoglycan, inner wall layer is peptidoglycan, which is surrounded by a mucilaginous sheath. Photosynthetic lamellae, thylakoids are present inside, which the pigments are present. Major pigments that are present inside thylakoids. Major pigments that are present include phycobilins rather than carotenoids and chlorophyll molecules, which are also present, but phycobilins are the major pigments that are present inside the cyanobacteria. Okay, so this is the cell structure and thallus organization of cyanobacteria. And then pigmentation. chlorophyll A, and majorly phycobilins. Phycobilins, which are water soluble pigments, from which phycocyanin emits the blue color. Okay, phycobilins which are water soluble pigments, amongst which phycocyanin will emit blue color and phycoerythrin will emit red color. Carotenoids will consist of beta carotene, which gives out orange color. Okay, beta carotene which gives out orange color, and chlorophyll B is absent. Fine, right? It has chlorophyll A, which conducts maximum amount of photosynthesis, phycobilins which are water soluble phycobilins like phycocyanin and phycoerythrin, and carotenoids which contains beta carotene. Okay, and chlorophyll B is absent in blue green algal members. There are some specialized cells that are also present, before which we will address reserve food material. What is the reserve food material that is present in blue green algae? Reserve food material is usually cyanophycean starch, which is of glycogen type. It contains cyanophycean granules. Cyanophycean granules are present in the cytoplasm, and it has oil droplets, and polyphosphate granules are present. Polyphosphate granules, which are very important. Now we will look into the specialized structures. What are heterocysts? Heterocysts are the enlarged cells with thick cell walls. Okay, these are the enlarged vegetative cells. See, if this is the filamentous form, few enlarged vegetative cells are addressed as heterocysts, and this heterocyst will have thick cell wall. Heterocysts will have thick cell wall with suberin depositions, which is impervious for gaseous exchange. This is impervious for gaseous exchange. Fine, right? This is impervious for gaseous exchange. And inside heterocyst, there is an enzyme present called as nitrogenase. What is the enzyme that is present inside the heterocyst? Nitrogenase. Okay. Nitrogenase is the enzyme which is present inside the heterocyst. This enzyme is for nitrogen fixation, and this will fix only during anoxygenic conditions. Anoxygenic conditions. Okay, nitrogenase will be inactive in the presence of oxygen. Okay, nitrogenase enzyme is inactive in the presence of oxygen. This enzyme will be active in the anoxygenic conditions. Okay, in anaerobic conditions. That is the reason the wall layer is very thick, which is impervious for gaseous exchange. So the anaerobic conditions are provided which will fix the nitrogen here. Nitrogen fixation. Fine, right? This can be terminal or intercalary. Heterocyst can be present at the terminal ends or it can be present in between. This is the major site for nitrogen fixation. This will lack photosystem 2. Photosystem 2 is absent here, whereas vegetative cells will contain both photosystem 1 and photosystem 2. Okay, vegetative cells contains both PS1 and PS2, photosystem 1 and photosystem 2, and pores will connect heterocyst with vegetative cells. What are those pores? These pores are known as microplasmodesmatal connections. Okay, microplasmodesmatal connections will connect in between heterocyst and vegetative cells. Heterocyst and vegetative cells. What is that connecting between these two? The pore-like structures which are known as microplasmodesmatal connections. Microplasmodesmatal connections. Clear with the heterosis? Now about akinates. What are akinates? These are thick walled resting spores. Okay, akinates are thick walled resting spores. Fine, right? These are thick walled resting spores which are rich in some reserve food material. They are formed from vegetative cells only. Okay, and they will germinate under favorable conditions. Fine, right? Whenever favorable conditions resume back, they will germinate into a new BGA. Fine, right? During unfavorable conditions, they will exist as thick walled resting spores. Fine, right? Any doubts till here, ma? I hope everything is clear till here. You are clear with the pigmentation, you are clear with special cells and reserve food material. Now about reproduction and movement. Before getting into the reproduction and movement, you should know about the cytoplasm which is present inside the BGA. Inside the blue-green algae, each cell cytoplasm is differentiated into peripheral chromoplasm and centroplasm. Okay, it has peripheral chromoplasm and central centroplasm. Peripheral chromoplasm will have pigmentation like phycobilins, which majorly consists of phycocyanin and phycoerythrin. Okay, which gives blue color and red colors, and it also it is predominant with chlorophyll A. Okay, chlorophyll A will give it green color and it will conduct photosynthesis. Clear, right? This is about peripheral chromoplasm, and central centroplasm will have the cellular constituents to run the metabolism. Now you see reproduction. We have already observed about the structures like heterosis for nitrogen fixation, akinates which aid in vegetative reproduction. Along with akinates, there is one more structure to be highlighted for vegetative reproduction, that is hormogonia. Okay, hormogonia, these are short motile filaments. Okay, hormogonia are short motile filaments which will help in the conduction of vegetative reproduction. Asexual reproduction is aided through akinates and endospores. Endospores is very rare. Okay, and movement is usually gliding movement, and as there is no flagella for BGA, usually gliding movement, like how a pendulum moves here and there, same gliding movement is observed for cyanobacteria. And then here we look into the ecological and economic importance. There is one more characteristic feature for blue-green algae which is addressed as Guidoco phenomenon. Guidoco phenomena, which means like usually blue-green algae will change its color according to the plant into which it is associated. Okay, so this change in color which gives camouflage activity is addressed as Guidoco phenomena, which is characteristic feature only for cyanobacteria. It will change its color according to the habitat in which it is living. Ecological and economic importance. First major importance is this is used as a biofertilizer. Okay, this is majorly used as a biofertilizer for nitrogen fixation. Second is this will increase the soil fertility. Soil fertility is increased. Third is this is used in paddy fields, especially Alocera and Oscillatoria. These are used in paddy fields. And then food by space travelers. Space travelers or astronauts will be using single cell proteins like spirulina and chlorella. Spirulina and chlorella, they will be used as SCP, single cell proteins, by astronauts for food purpose. They are used as food supplements. Oxygen production. Water blooms are formed. Water blooms are formed by some parasitic cyanobacteria like Microcystis. microcystis, which forms water blooms. And then toxin production. Toxins will be produced by Anabaena and microcystis. Clear? These are some basic features. Fine, right? Any doubt anywhere, ma? Now, we learn 3 type studies in comparison. Okay, one is nostoc, second Anabaena, third oscillatoria. We will learn it in detail in comparative analysis. So first you look into the schematic position now. Of course, all of them will belong to the same class, that is Cyanophyceae, Nostocales and Cyanophyceae. Here also the same. But oscillatoriales, okay, this will be the family for oscillatoria. Habit, if you see, what type of habit, what type of structures they possess. Nostoc, it can exist in filamentous, collinear or gelatinous form. Anabaena exists in filamentous and free floating form. Oscillatoria exist in the form of filamentous and free floating. Fine. Now, habitat, fresh water, mostly soil, and it exists in symbiotic association with Anthoceros, Cycas and Genera. Okay, with these plants it will exist in symbiotic association for the purpose of nitrogen fixation. Anabaena, usually it is a fresh plankton and it is also a symbiote. It will have a symbiotic association with Azolla. Oscillatoria is usually found in freshwater habitats like ponds and lakes. Next, nature of thallus. Nostoc has jelly-like colony which is which contains filaments. Anabaena is single unbranched filament. Oscillatoria is single long unbranched filament. Colony, here you see, this is present in gelatinous form. Colony is absent in oscillatoria as well. Trichomes. Many trichomes are embedded in mucilage sheath. Trichomes are surrounded by mucilageous coat all around the nostoc. Okay. What are trichomes? Single row-like structures which are emitting out. Whereas if you see for Anabaena and oscillatoria, there is just single trichome for both of them. Okay. Next is cell shape. Cells are usually spherical or spherical to barrel shaped. Here Anabaena cells are bead-like. Oscillatoria cells are disc shaped. Okay, oscillatoria cells will be disc shaped. And then arrangement of cells. How cells are arranged? Cells are arranged in series within the trichome. Here cells are arranged in the form of chain. Okay, cells are arranged in the form of chain. And here how they are arranged? In a complete linear row. Next, prokaryotic nature is present in nostoc, Anabaena and oscillatoria because they are having what cells? Prokaryotic cells. There is no proper membrane bound nucleus. Nucleus is absent in all of them because they are what? Prokaryotic cells. Cell nature is prokaryotic. Next is about pigmentation. What are the major pigments that you observe? Nostoc, it is chlorophyll A, majorly phycocyanin, phycoerythrin, carotenoids. These are phycobilins. Where are they present? Same as that of nostoc. All the pigmentation is present in the peripheral chromoplast. They are present in peripheral chromoplasm. Fine, right? Fine. Next, chlorophyll B is absent in all of them. Only chlorophyll A is present. Remember, which is a major point. Whenever pigmentation type of questions are asked regarding pigmentation of cyanobacteria, if at all chlorophyll B option is given, you have to eliminate that. Okay, clearly remember the pigmentation. Next is reserve food material. In nostoc it is cyanophycean granules. You can also call it as cyanophycean starch oil and volutin. Here also it will be the same. Next is heterocysts. Heterocysts are present in the nostoc, can be intercalary in between the vegetative cells or at the terminal or apical region as well. Whereas in Anabaena, it is strictly intercalary in position, whereas in Oscillatoria, the heterocysts are absent completely. Next is function of heterocyst. Of course, it is nitrogen fixation. Remember, heterocysts are absent in Oscillatoria. So, there is no function here about heterocysts. Remember that carefully. Next is about echinates. Echinates will be present in nostoc and Anabaena. Again, in Oscillatoria, they are absent. Next, gas vacuoles. Gas vacuoles are present in Anabaena and Oscillatoria. In both of them, gas vacuoles are present. Next, what is the type of branching that you see? There is no branching in nostoc, Anabaena and Oscillatoria. Trichomes are single rows only. Next, movement. Movement is gliding or slow movement. Here also it is gliding movement. In Oscillatoria, we can see three types of movements. One is like pendulum, second is like rotatory movement also is observed. Okay, like how a pendulum moves here and here. Same ways it will be moved. Fine, right? Okay, that movement is known as oscillatory movement. That is why the name is given as Oscillatoria. Next, vegetative reproduction is by fragmentation and hormogonia. Echinates and hormogonia. Here it is only hormogonia. See, there are clear differences between nostoc, Anabaena and Oscillatoria. Please remember, Oscillatoria is more variable when compared to Anabaena. Fine, right? Next, sexual reproduction is absent in all of them. Special structures. What are the special structures that you can see? In nostoc, gelatinous sheath is observed, heterocysts are present and echinates are formed. Anabaena, if you see, heterocysts, echinates and gas vacuoles are present. Next, Oscillatoria will have cross walls which are visible and there is no heterocyst at all. Okay, there is no heterocyst and cross walls will be visible here in the Oscillatoria. Clear? Next. Yeah, last set of differences between these. Nostoc, Anabaena and Oscillatoria. Last set of differences. Nostoc, Anabaena, Oscillatoria. Here we go. First is about symbiosis. With Anthoceros and Gunnera, this shows symbiotic association and the major purpose of symbiotic association is nitrogen fixation. Next is with Azolla, Anabaena associated with Azolla. Here it is absent. Nitrogen fixation is present in nostoc and Anabaena, absent in Oscillatoria. Water blooms formation is rare in nostoc. It is common in Anabaena and Oscillatoria. Economic importance, if you see, soil fertility and nitrogen fixation. Here in Anabaena, this acts as biofertilizer, where Anabaena is associated with Azolla. Oscillatoria, oxygen production is observed. Fine. What is the negative role of the nostoc? These are rarely toxic. They form toxic blooms. Oscillatoria also forms toxic blooms in water bodies. Characteristic feature is jelly-like colony. Anabaena is Azolla symbiont, and here characteristic feature is oscillatory movement. Okay, oscillatory movement which is observed. So with this, complete cyanophysia, general characteristic features and 3 type studies, nostoc, Anabaena and Oscillatoria are learnt in comparative analysis. And now we will move on to mycology. Okay. What are the concepts that we are going to cover here first? In mycology, what are the concepts that we have to cover? Look at this carefully. First, here you see, of course, general characteristics of fungi. First we will finish off this. Next, substrate relation in fungi. We will get exposed to many different types of hyphae and mycelium, like primary mycelium, secondary mycelium, septate, aseptate type of mycelium. So we will get exposed. So all substrate relationships in fungi we will get into. And most important, cell ultrastructure, and fungi is eukaryotic cell. Fine, right? So cell ultrastructure we will see, unicellular and multicellular organization. By type studies we will learn, like unicellular Saccharomyces cerevisiae, multicellular mushrooms we will learn in detail. Okay. Cell wall composition. Cell wall is made up of chitin which has NAG units inside it. Okay. And then we will look into nutrition. Nutrition is not diverse in fungi. It is purely heterotrophic nutrition. Nutrition is heterotrophic. Fine, right? Nutrition is completely heterotrophic mode of nutrition. This heterotrophic mode of nutrition can be saprobic, biotropic and symbiotic. 3 modes of heterotrophic nutrition. And reproduction can be vegetative, asexual and sexual mode of reproduction. And then we will look into heterothallism. Heterothallism, where we will highlight about primary and secondary myceliums. Next is heterokaryosis and parasexuality, followed by molecular aspects involved in classification. What are the molecular aspects involved in classification? And then about clear characteristic features of mastigomycotina, zygomycotina, ascomycotina, basidiomycotina. Fine, right? Where after learning each characteristic features under each class, we will be learning about the type study under each class. Under mastigomycotina, we will be learning about albugo, zygomycotina mucor, ascomycotina Saccharomyces unicellular form, penicillium multicellular form. Basidiomycetes, which is the advanced class of fungi, we will be looking into Puccinia. Then Deuteromycetes, Sarcospora, which is parasitic fungi, again we will learn it in detail. Next, about economic importance of fungi and toxic fungi which are causing diseases. When we are going through the general characteristic features, reproduction is the most highlighted topic of fungi. Okay, reproduction is the most highlighted topic. In this reproduction, we have to focus both on the asexual spores.
We will consider, right?
And sexual spores. How they are produced? Both exogenous and endogenous mode of release. Fine, right? These are the characteristic features under mycology. Clear with it? Any doubt anywhere? And then we will move on to the introductory point. Fungi general characters and classification. What is the nature of fungi? Fine. First, it is highly specialized, non-chlorophyllous, heterotrophic fungi. Okay. Highly specialized, non-chlorophyllous, heterotrophic organisms. Fine. And they are eukaryotic organisms. Fungi will possess long thread-like structures. These long thread-like structures are addressed as hyphae. Each cell is surrounded by a cell wall which consists of chitin. Here a bit we will move on to the classification once, where we will also learn it in taxonomy. As it is having cell wall which is the major criteria for two kingdom system of classification. In two kingdom system of classification by Carolus Linnaeus, fungi is kept under plant A because it is having cell wall. Then under five kingdom system of classification, when mode of nutrition is the major criteria, it is again put into the separate kingdom by R.H. Whittaker. Remember this point can be addressed. Okay. Reproduction is both asexual and sexual way, and it is through what? Spores. All the asexual and sexual mode of spores, their mode of release you have to remember. Next, types of fungi which is based on the form. We have macroscopic forms. Macroscopic forms include mushrooms, puffballs and bracket fungi. These are the macroscopic forms. Filamentous forms include Aspergillus penicillium. These are the filamentous forms of fungi. Next, unicellular forms include yeast, which is Saccharomyces cerevisiae. Fine, right. What is etymology or mycology? Etymology or mycology is one and the same, which is nothing but study of mushrooms. Okay, it is study of mushrooms. Fine. Next, in Greek, mycos means mushrooms and logos means study. So it is, these are the words which are derived from Greek. That's it. This is basic introduction of mycelium. Historical milestones that we have to remember about scientists. Just go through this, you can easily get into. Purely memory based. Scientists and their discoveries of fungi. Occurrence of fungi. Fungi is ubiquitous, it is present everywhere, ubiquitous in its distribution. Okay, fungi is ubiquitous in its distribution. It is widely distributed. They are found on the substrates with organic matter and moisture. Okay, and they lack chlorophyll. They don't have chlorophyll, they are achlorophyllous. Hence they are heterotrophic in nutrition. Okay, they are heterotrophic in nutrition. Okay, just a second. They are heterotrophic in nutrition. Most fungi are saprophytic. They grow on dead organic matter. Some fungi will be parasitic. They cause various plant diseases. Some fungi will also show symbiotic relationships. Okay, like fungi along with algae, they form what? Lichens. Fungi with roots of the higher plants, they form mycorrhiza. Fungi with roots of the higher plants, they form mycorrhiza. Fine, right? This is symbiotic association. One example with roots of higher plants, if I have to tell you, pinus. Fungal association with pinus. Pinus roots. Okay, there are two types of associations. It can be ectomycorrhiza and endomycorrhiza. Ectomycorrhiza and endomycorrhiza. Take the root system.
So
Science is having what type of root system? Tap root system.
The step first step to you need to for this question.
So this taproot system, if fungi is associated from the outer, if fungi is associated from the outer regions like this,
N2 is 0.1, area is 0.0.
this is ectomycorrhizal association. The main
End to end, sorted end.
association here is to increase the surface area of roots, thereby increasing the absorption of water and minerals. Okay, primary purpose is increase surface area of roots, which thereby increases the absorption of water and minerals. Fine. And then here you can see ectomycorrhiza. Endomycorrhiza will dwell into the walls inside the cytoplasm. It will go. Okay, the mycorrhizal associations or the fungal association will go into the cells and they will build connections. Intracellular connections will be built. Such type of mycorrhiza is known as VAM, which is vesicular arbuscular mycorrhiza. Okay, this is vesicular
3 important third
arbuscular mycorrhiza. Clear till here, ma? Any doubts anywhere? Fine. Now, what is the status of fungi? Just now we have once recalled about 2 kingdom and 5 kingdom system of classification. Here, Whittaker in the year 1969 has proposed 5 kingdom system of classification, and fungi being purely heterotrophic nutrition and thalloid type of body organization, these are put into a separate kingdom. Separate kingdom of fungi is given by R.H. Whittaker in 5 kingdom system of classification, where we have 5 kingdoms: Monera, Protista, fungi, Plantae and Animalia. Okay, we have Monera, Protista, fungi, Plantae and Animalia. Fine. Elevated fungi to separate kingdom in 5 kingdom system of classification. Clear till here? Any doubt anywhere, ma? Yeah. And then Cavalier and Smith, they have proposed 2 empires and 6 kingdoms. Those include Prokaryota and Eukaryota. Prokaryota includes 2 kingdoms, Archaebacteria and Eubacteria. Eukaryota includes 6 kingdoms again, which include Archaeozoa, Protozoa, fungi, Chromista, Plantae and Animalia. First 2 kingdoms, Archaeo and Protozoa, are lower level with simple body organization. Rest all are of higher body organization. Fungi is considered as a separate kingdom on what basis? First is nutrition. Okay, nutrition in fungi is purely heterotrophic nutrition. This heterotrophic nutrition, it can be saprophytic, mostly it is saprophytic, at times parasitic and symbiotic. Next, storage food material will be glycogen, biochemistry, cell wall composition, cell wall chitin, which is also,
and this is 1 right? No, so 79.
this is also addressed as fungal cellulose. They show varied growth patterns, hyphal structures are observed, and
Or I can take
also look into genetics.
1 or
Looking at
to other side, or
fungi is
in this thing.
into a separate.
we can divide.
Clear till here? Any doubts anywhere, ma? And then about mycelium structures. Fungi will have 2 types, 2 forms of fungi are observed. One is hyphal forms, which are the filamentous forms of fungi. Next is unicellular forms, hyphal forms and unicellular forms. Fungal body is made up of hyphae, which is called as mycelium. Okay, fungal body is made up of hyphae, which is addressed as mycelium. Majority of fungi are mycelial forms. Most of them are mycelial forms, and they are referred to as what? Molds. They are referred to as molds. Next, types of fungal structures. Yeasts are usually unicellular forms. Yeasts are unicellular forms. Apart from hyphal forms, we have 2 additional forms like mycelium with rhizoids and dimorphic fungi. What is dimorphic fungi? Single fungi, single mycelium structure or single fungal body has two structures. What are those two structures? Primary structure and secondary structure, which is primary mycelium, and secondary mycelium. What is this primary mycelium and secondary mycelium? Primary mycelium is the fungal body before giving rise to the spore. Okay. Secondary mycelium will give rise to
bud. Okay, you can take a look of the
fruit.
We can go for the next question.
Find a single fungal mycelium which consists both primary mycelium, which is the primary mycelium structure, and secondary mycelium which has fruiting bodies that releases out spores, is known as secondary mycelium. Both primary and secondary mycelium, if they are present in the same fungal body, it is known as what? Dimorphic fungi. And then in unicellular fungi, cells can be spherical, ellipsoid, tubular or regular in shape. If entire thallus transforms to a reproductive structure, this is called as holocarpic structure. Okay. Dimorphic, which has both primary and secondary mycelium.
Cross section A1 and A2. L1, L2 both are equal. Cross section area A1 and A2 they have given. Okay. Have their ends at the same temperature. Ends at the same temperature they have given. No. T1 and T2. And K1 and K2 are the thermal conductivities of two rods. So two rods they have taken as, and the rate of flow of heat in is same in both the rod, if they are asking. Okay, two rods. So this is how much K1 and this is K2. So Q by T will be equals to K1 A1 delta T divided by L1, right? And when we talk about the thing, they have given that equal K2 A2 delta T divided by L2. So K1, L1 and L2 are equal, I can cancel it out. A1 and A2 they have given, K1 and K2 they have given. Delta T is also same, like temperature is same. Delta T cancel it out. K1 A1 will be equals to K2 A2. Option K1 A1 equals to A. Yeah, they have given in terms of like this, A1 divided by A2 will be equals to K2 divided by K1. A1 divided by A2, K2 divided by K1. Option B is the correct answer. Option B is the correct answer for this question. Okay, I guess hope clear. Next question, see here, two identical vessels has been given, made of different materials, different material in the sense itself, K1 and K2 will be different, right? Yes, are completely filled with ice that at 0 degree Celsius. Due to temperature of a surrounding, the ice of a two vessels melt 25 minutes and 20 minutes. Here is the thing, why I have given the value H as Q by T, understood? Then the ratio of K1 and K2. So when we talk about the rate of heat conduction through the vessel, Q by T is equals to Ka delta T by L. So when we talk, Q by T is directly proportional to K, directly proportional to L, inversely proportional to, directly proportional to A, K, and inversely proportional to L. And when we talk about the thermal conductivities and the time, so 1 by T is directly proportional to K. So T1 by T2 equals to K2 by K1. What is T1? 25. What is T2? 20. So 25 divided by 20 equals to K2 by K1. So K1 by K2 will be equals to 4 by 5. 4 by 5. Option B is the correct answer. Option B is the correct answer. Done? Okay. So the next question. Two rods A and B are connected in series as shown in the given figure. The conductivity of A is K1, that is 100, and the conductivity of B is K2, that is 50. Free end of a rod A and B as a temperature 30 and 60 degree respectively. Temperature of the melting point will be. So what you do is reverse this as it is. Reverse this. Or what is the thing here? The temperature is increasing. No, temperature is increasing. In the sense, like if you come from this side, temperature is decreasing. No, so take B and A. B equal to A like this. So KB AB delta T divided by LB will be equals to KA AA delta T divided by LB. So LA. Right now, what is KB? K2. 50 they have given. 50. And what about the area? Two rods. Okay, A1, A2. And if you talk about the area respectively, they have given the area. Connected series. The free end of a rod A and B, temperature, common melting point. Okay, I guess area is missing, nah? Area is missing. Okay, we'll keep that area as it is, we'll see. So this is area AB and the temperature 60 minus T divided by the length, how much? 2 will be equals to, and this is how much? This how much? How much this is? 25 or 100? 100 they have given, 100, and this is AA, this is T minus 30 divided by 1. 1 times, and this is 2 times, and this is AB 60 minus T is equals to 4 AA T minus 30. They have given area, I guess. Wait a minute. Okay. So we'll take the area. I'll give the area for this question. You can take the area. Okay. Connected is AB is free and A and B 30 degree and 60 degree respectively. Melting point P. Okay. Let area of A1 be 2, area of A2 be 4. Okay, it's a series. No, okay, it's a series thing, nah, it's series. When we have talked about series, if you see, yeah, there is no need to talk about the area. Area is same in series, right? Yes, it's a series thing, nah. We were discussing about series only, no. So this A and this A will get cancelled off. 60 minus T will be equals to 4T minus 120. So 120 take this side.